Okay, after looking at how transcription is initiated, right, we look at the next stage of transcription, which is transcription elongation. So the process of elongation is the same between pro and eukaryotes, right? So we look at them together. Okay, so at the end of transcription initiation, right, we call that the transcription bubble is formed. The DNA double helix start at the gene, right? The start of the gene is unwound, right? So the template strand and the non-template strand are separated. Okay, so when the template strand is now exposed, the nitrogenous bases are exposed. Free RNA nucleotides in the nucleus or in the nucleoid region, if it's a prokaryote, the free RNA nucleotides can now bind to the nitrogenous bases of the template strand via complementary base pairing, right? So if it is an A on the template strand, then a U RNA nucleotide would bind. If it's a C on the template strand, then a G would bind, and so on. All right. So once the free nucleotides have bound onto the template strand, the RNA polymerase would catalyze the formation of phosphodiester bonds between adjacent RNA nucleotides. Okay, and that would synthesize the mRNA, and as the RNA polymerase moves along the gene sequence, move along the template strand of this gene sequence, then the mRNA transcript is elongated. Right? The elongation happens in a particular direction, in the 5' to 3' direction with respect to the growing mRNA. Right? So since nucleotides are always anti-parallel, for the template strand, it would be running from 3' to 5'. Right? So the RNA polymerase would move along the template strand in the 3' to 5' direction with respect to the template strand in order to elongate the mRNA in the 5' to 3' direction with respect to the growing mRNA. All right. So just to look at more closely the binding of the free nucleotides, as you can see, right? The free nucleotide comes in. The free nucleotide is always in the form of a triphosphate, right? So it's a particular nitrogenous base nucleoside triphosphate. Right? So for example, the adenosine triphosphate, a cytosine triphosphate, and so on. Right? So the free nucleotides are in the form of a triphosphate. It will bind to the exposed template strand. This is the exposed template strand here via complementary base pairing. And then the RNA polymerase would catalyze the formation of the phosphodiester bond right here, right? Phosphodiester bond between two adjacent RNA nucleotides. Right? And as the phosphodiester bond is formed, then two of the phosphates get released. Okay, so this is the process. Once unwinding has happened, right? Once the template strand is exposed, again complementary base pairing, so that the free RNA nucleotides can bind, and then the catalysis of the phosphodiester bond by RNA polymerase. So this is the growing mRNA strand, right? So the mRNA strand will grow from 5' to 3', right? This is 5' at the start, right? Then 3'. So at the end, it will be the 3' here, right? So 5' to 3' direction. Okay. Right. So as the elongation continues, right? If you look at the bubble, yeah, you see that the bubble kind of shifts along the DNA molecule, right? So this is a bit different from DNA replication, if you recall. For DNA replication, right, the bubble would continue to expand, right? If you remember, right, unwinding happens in both directions in DNA replication. So the bubbles get bigger and bigger. But for transcription, it's different. For transcription, right, the RNA polymerase unwinds the DNA double helix, so transcription can continue. But once transcription is done, right, at the end, at the back here, at the back of the RNA polymerase, once transcription is done, the DNA actually rewind. So the DNA don't unwind in this direction. The DNA actually rewind, right, after the RNA polymerase has left. Okay, so but as RNA polymerase continue to move, right, upstream here, up ahead here, right, the DNA will be unwound by the RNA polymerase, but at the back, once the RNA polymerase has moved past, the DNA actually rewind, and the mRNA strand, right, is left like kind of dangling, right? It peels away. That's what I've written in the notes. It peels away from the DNA, right? So it exists as this single-stranded RNA transcript here that is continuing to be elongated. All right, so the transcription bubble kind of moves along, right, as it unwinds ahead and rewinds at the back. It just moves along the gene sequence all the way to the terminator sequence, right? Okay, so this is a little bit different from the process of DNA replication. Okay, so the RNA transcript, right, again, would be elongated in the 5' to 3' direction as the RNA polymerase moves along the template strand in the 3' to 5' direction. Okay? All right, so if you understand that, right, then here is an exercise, right, to just consolidate and also to make a comparison with the process of DNA replication. Right, so spend some time to try out the questions here. Okay, you can pause the video, and once you're done, you can then start the video again, right? Okay, so if you are done, right, then I'm just gonna go through the answers with you. Okay, so for the function of the two enzymes, RNA polymerase for transcription and DNA polymerase for DNA replication, they have different substrates. The substrates here refers to the raw materials, right, to build the newly synthesized nucleotide. Right, so for polymerase, RNA polymerase rather, for RNA polymerase, it synthesizes a mRNA sequence, right? So obviously the substrate would be RNA nucleotides. For DNA polymerase, it synthesizes a daughter DNA strand, right? So obviously the substrate would be DNA nucleotides. Right, products I mentioned just now, mRNA versus a daughter DNA molecule, right, because it builds the DNA strand, it synthesizes the daughter DNA strand, right, and the DNA strand would be bound onto the parental strand, and that forms a daughter molecule, right, and there's no need for a free OH for RNA polymerase, but there's a need for a free 3' OH provided by the primer for the DNA polymerase function. Okay, similarities, right? They both catalyze phosphodiester bond between adjacent nucleotides. Of course, the nucleotides is different. One is RNA nucleotides, the other is DNA nucleotides. But in terms of formation of bonds, it's the same bond. Right? The direction of elongation is the same as well, right? So both elongates in the 5' to 3' direction. Right? Both requires a template, and both involve the formation of the same kind of bonds, phosphodiester bonds. Okay? Yep, so just a consolidation there. All right.